Slide. So now we are going to start this hands-on session on microscopic imaging to classify normal and abnormal pancreas cells with sequential convolutional neural network. So I think with me today, Jagrita Pal, one of our research scholar of computer science and engineering department, and I think basically she will explain VGG, uh, VGG, that is called Visual Geometry Groups Deep Neural Architecture. She will describe today, and it is, I think, very valuable talk, and some programs, how to write the program, and some demonstration. So it is developed by the University of Oxford, the discernment team in 2014, I think. So I'm requesting Jagrita to start this session.
So thank you, sir, for giving me the opportunity to present today's tutorial. Good afternoon, everyone. Myself. Myself, Jagrita Pal, a research scholar from the Department of Computer Science and Engineering, Tripura University. So today my topic will be hands-on practice using the microscopic images to classify normal and abnormal precancerous cells with sequential convolutional neural network. Proceeding towards my first slide. So these are the contents which I have included in my presentation today. So first of all, in the introduction part, I'll be giving a little explanation about what is cell and what is the need to do image classification, what is its importance, and what is the need for microscopic image classification also. Although it has been explained earlier, so I'll be giving just a few concepts about that. And then what are the different types of image classification and what datasets I'll be using for microscopic image classification. That is two datasets, AGMC2 dataset and high-level dataset. And the model which I'll be using for image classification is VGG16. And why I have used particularly VGG16 for image classification, I'll be also explaining that what are the basics of VGG16 and that also the diagrammatic representation of microscopic cell image classification on AGMC2 dataset. And what are the data augmentation techniques and the Python code running platform for AGMC2 dataset as well as the high-level dataset, because I'll be explaining about the two datasets. So yeah, and then conclusion and references. So this my introduction, like what is a cell? As you already know, cell is the basic unit of life, I guess. So a cell is the structural and functional and biological unit of an organism. Cells are the basic building blocks, and human bodies are composed of cells, as Madam already, Professor Niharika Nath Madam already explained what is cell, and you all know about that. So I just kept a basic concept what is cell. Every cell is composed of plasma membrane, cytoplasm, nucleus, and a nucleus or whatever. And in eukaryotic cells, it is known as nucleus, and in nuclear in prokaryotic cells. So basically, I'll be performing image classification, that is microscopic image classification in cells. So what is the need for image classification? Image classification is a fundamental task in computer vision, which involves assigning a particular category or label to an entire image based on visual content. Like there are various class levels in an image. If we want to classify each class level in different way, then we need image classification to enable computer see and understand images by assigning them to predefined categories. Like I have taken an example here where example in that example, like there are various figures like a cat, dog, and a frog, and if we train the model, like it is a supervised learning technique where we will be giving the labels as well as the images to the model to train them so that it can classify which one particularly describes which part. So here, the model after training the model, it clearly classifies which one is dog and cat. It is just an image, nothing else. Classification of an image using machine learning. Proceeding towards next slide. Why do we need microscopic cell image classification? So basically, I'll be doing microscopic cell image classification using a deep learning neural network. So why should we do that? The need for microscopic cell image classification arises from its crucial role in automating cell-based analysis for applications like disease diagnosis, drug screening, and genetic research. Generally, microscopic image classification plays a vital role in biomedical research and healthcare, as it allows models, computational models, to automatically analyze several cells and identify shape, textures, and patterns. Traditionally, this process is done manually under a microscope, which can So the types of image classification are, depending on the problem at hand, there are different types of image classification methodologies, which are screenshots. So it is a technical issue. I'll be continuing from that part. Depending on the problem at hand, there are different types of image classification methodologies that need to be employed. So the datasets I have taken is a GMC2 dataset and HighLev dataset, which I have told in the content part. So these are generally binary classification dataset and multi-class classification datasets. What is binary classification generally? Binary classification takes an either logic or two label images and classifies unknown data points to two categories. Like when our task is to categorize benign and malignant tumors, analyze product quality to find out whether it has defects or not, and many other problems that require yes or no answers are solved with binary classification. For example, if we take a dataset, that is the dataset which I'll be working on, a GMC2 dataset, that is a binary class dataset. So in binary class dataset, there are generally two classes. That is the dataset which I'll be working on has two classes, abnormal and normal. cancerous cells and normal cancerous cells. So when there are two classes from a data set, that time we can use a binary classification technique. In case of multi-class, that is more than two classes in a dataset, that time we can call it as a multi. I will be working on from next slide, upcoming slides. So
Hello participants, wait for 5 minutes, there is a technical issue for the network issue. Wait.
so am I audible? Am I audible?
Yes, Jagrita, please carry on.
So multi-class classification, as the name suggests, categorizes items into three or more classes. As I have already said, more than two classes could be considered as multi-class classification. Like here in the image, you can see that there are three classes of so the network is a bit disturbing today. So I'll be continuing from this part. Here you can see that there are three classes which are classified as multi-class classification. Basically, machine looks at the features, like how is it classified in different, how is it classified in different way. Basically, the machine looks at the features like what is that shape, color, texture. So while the model is being passed through neural networks, so the machine generally identifies the particular shape, color, or texture and chooses in which class the picture is most likely to be based on the training it received. Proceeding towards next slide. What are the datasets I took for microscopic image classification? Here we'll be performing image classification for microscopic data, where basically we consider two datasets. As I have already mentioned in the content part, I'll be taking the AGMC2 dataset and the HiRLEF dataset. For AGMC2 dataset, it is a binary class dataset where there consist of two classes, that is abnormal image, abnormal cancerous cells and normal cancerous cells. So I'll be training my VGG16 model on that on that data so that it can classify the cancerous cells as abnormal and normal correctly. So for the AGMC2 dataset, a binary classification task is performed since it contains two categories of cell images. In contrast, the HiRLEF dataset is a multi-class classification task. It comprises of 7 distinct classes of cervical cell images. In HiRLEF dataset, the class consists of 7 distinct class features, like there are 7 different class categories. So I'll be performing the my I'll be training my model on HiRLEF dataset too, so that it classifies particularly an image belongs to which particular class. In this project, I have utilized the VGG16, which is a deep learning model for microscopic cell image classification. So why I have taken VGG16? VGG16 is a convolutional neural network, that is CNN architecture, which is specifically designed for and widely used in image classification tasks. It is one of the popular algorithms for image classification and is also easy to use. There are various other classification architectures. There are various other classification architectures also. But the main reason why I chose VGG16 is that it offers really a good balance between accuracy, simplicity, and computational efficiency. Since VGG16 has slightly fewer layers as compared to other classification architectures, so it almost equally trains faster, especially on smaller data sets. It is also a much less complex model as compared to other algorithms or other neural network architectures, so I've preferred VGG16 over my dataset. For my dataset and experimental setup, VGG16 provided excellent performance with less complexity. We have used the supervised learning technique here, where I'll be giving, as you all know what is supervised learning technique, we'll be training the model by giving the input images as well as the levels, we'll be passing the input images to the VGG16 model neural network architecture, so as the model to learn about the features, about the shapes and textures. We have used the supervised learning technique here using the pre-trained model weights of VGG16. Overall, VGG16 was the most practical and efficient choice, offering high accuracy with simpler implementation and faster training time. So we'll be explaining about the VGG16 model from the next slide onwards. But to understand the basic concepts of VGG16, we need to understand about what is CNN, like what is convolutional neural network. I just given a few topics about that. Like here I've taken an image, so this is just a small part of that image, which is also can be called as a patch. So and generally an image consists of pixel values. Like if we make it enlarge it, then it considers of just numerous numbers, numeric values. Imagine you have a photo and you can tell it's a cat, but how can a computer recognize it? A computer only sees a grid of numbers, that is the pixel values. What the computer sees, that is, after training the model, it can classify as what is it and what is not. So we build a model that can learn to see shapes, edges, textures, just like our eyes. And we can say that as convolutional neural network. Here the image is three-dimensional arrays of integers ranging from 0 to 255. As you all know, the pixel values come from 0 to 255 and of size, width, height, and 3. Here the 3 represents the RGB, that is red, green, and blue, 3 color channels. So it's just an image, nothing else. That is training a model about classification, like what are they giving different levels and training it, which are called oranges and what are called apples. So if we give the input as that, so it can easily classify that there are 2 apples in the picture. VGG16 is one of the most famous convolutional neural network, and simple, deep, and powerful. Proceeding towards my next slide, the VGG16 model is a convolutional neural network. Like now we are going through the basics of VGG16. Who proposed that for the first time? That was proposed by the Visual Geometry Group. That is the full form of VGG, Visual Geometry Group. Sir, I've already mentioned earlier what is the full form of VGG, that is Visual Geometry Group. And it was first proposed by them at University of Oxford in the year 2014. So the paper name was Very Deep Convolutional Neural Network for large scale image recognition. I have already mentioned it in the references, so you can visit there in the reference part. So it is there in 2014. This paper was first proposed by them. So this is the overall architecture of VGG16. Like first, this is an abnormal or normal. This is a cancerous cell. This is the image I have taken here is a cancerous cell. So here the image height, here 224 represents the image height, and this 224 represents the image width, and 3 is the RGB color channels. So while passing the image as an input to the convolutional neural network architecture, here these two convolution block. It represents already, I have mentioned which block represents what. Here convolution and these blocks, and then here will be performing max pooling. What are the operations I have explained elaborately on the further slides. So first there are two blocks of convolution and then max pooling happens. So what is the work of max pooling and how convolution happens? I have elaborately explained the mathematics in the further slides, and how the width, image height width reduces, and generally 64 is the number of filters. So how 64 increased to 128 and 256, I will be explaining all over that. So convolution, and what is ReLU function, how that works, what is max pooling, and I'll be explaining all over that. So convolution 1 has 64 number of filters, that is, these are the number of filters. So I have considered convolution 1 and convolution 2, I mean both, taking both, I have considered this is a single block of convolution 1, and convolution 2 has 128 filters, convolution 3 has 256 filters, as well as convolution 4 and 5 have 512 filters. So this is the architecture. Um, yeah, VGG16 architecture. What does the 16 in VGG16 refers? So it refers to 16 layers that have weights. Here, just count the number of layers here, 1, 2, and we'll be not considering the pooling layers because I have also explained this part. In VGG16, there are 13 convolution layers. As you can see here, you can count the number of convolution layers that sums up to 13, and 5 max pooling layers and 3 dense layers. The dense layers are also known as fully connected layers, so which helps in decision making. What is the work of dense layer? Mainly, it helps in decision making that the object belongs to which class while passing an image, that is a particular object in the image belongs to which class, that is mainly determined by the dense layer. Which I mean, if we add all the layers, then it sums up to 21 layers. But why it is called 16 then? It is only 16 weight layers, that is learnable parameters layer, something that the neural network adjusts during training to minimize the loss function. Now, non-learnable layers are pooling and the dense layers. That is, it doesn't consider any weights, and the weights are not increased or multiplied during the operation. So they are called non-learnable parameters or non-learnable layers, like pooling. Don't have any weights or biases. They only perform fixed operations, so they are not considered as any learnable parameter. Here, 5 max pooling layer just reduces the spatial size. What is the work of max pooling? Just it reduces the spatial size of the image. As I have already explained while while going through the convolution architecture, that is VJ16 architecture, the image size reduces. How it reduces? Because of pooling operation, it takes the maximum value. I will be explaining that on next slide. 5 max pooling layers just reduces the spatial size, which refers to physical dimensions or extent of an object or phenomenon, including its length, area, or volume. The convolution and max pool layers are consistently arranged throughout the whole architecture. There is convolution layer and max pool layer are arranged through the whole architecture. VGG16 takes input tensor size as 224 cross 224 with 3 RGB channel. As in the previous slide, we already saw that how it takes the input image size, that is image height would be 224, image width should be 224, and with 3 RGB channel. If we look at the number of filters that we can use, around 64 filters are available that we can double up to 128 and then 256. So why do we double the filters? Just so that when the image size reduces to particularly specify the texture, shape, and the region of interest, we generally increase the number of filters so it gives better output. In the last layer, we can use 512 filters. So proceeding towards next slide. VGG16 is also called a sequential CNN architecture because its layers are stacked one after another. Why VGG16 is all sequential? Since in my topic was that I will be using a sequential convolutional neural network. So why VGG16 is called a sequential CNN architecture? Because its layers are stacked one after another in a single linear chain, with each layer having one input and one output. Like you can see that I have added the previous image only here. Here, each of the layers are stacked one after another in a linear chain, like linear way. You already know what is meaning of linear, like one after another. Here, each layer have one input and one output. Like when we'll be giving for convolution 1-1, there is one input, and the output from convolution 1-1 will be the input of convolution 1-2. That is, every layer has one output and one input. So linear stack of layers in a sequential model, the output of one layer becomes the direct input of next layer. Here, the convolution, the output of one layer becomes the direct input of next layer. VGG16 follows this by arranging 13 convolution layers and 3 fully connected layers in a single line. Here, VGG16 arranges it like there are 13 convolution layers which are arranged in a single line. Like there are not any extra multiple inputs or outputs. It's just like any input from this side and it goes through in a serial manner or like in a stack. So this is also called as going by sequence. So it is also known as sequential convolutional neural network. No multiple inputs or outputs. This sequential arrangement means the model is not designed to handle multiple inputs or outputs simultaneously. That is, this sequential arrangement is not designed to handle multiple inputs. If you're giving multiple inputs, that it will give a error. Like this model is not designed for multiple inputs. So data moves forward in one path from start to finish, which is ideal for simple image classification task. For simple image classification, data just moves in one path, like there comes one input and then it gives output, and the input layers goes through this, and finally we get the output. That's why it is known as sequential convolutional neural network, because the whole architecture is just comes in a sequence, that is in a linear manner. So this is the diagrammatic representation of microscopic cell image classification on my AGMC2 dataset. So as I have already mentioned earlier, AGMC2 dataset is a binary classification dataset where it consists of only two classes, abnormal cancerous cells and normal cancerous cells. We'll be passing both, both these, here, this is the collection of AGMC2 dataset for binary classification, where inside it we'll be passing normal, these are the normal microscopic cells. As you can see, as you have already heard from previous lectures, that the nucleus, in maximum cases, and the nucleus remains small for normal for microscopic cells, so you can already identify that as normal classification, and for abnormal, the nucleus is much more larger. So you can already say that as normal, and these are some of the 4 images. Normally in AGMC2 dataset we have 50 images for both abnormal, and one for normal there are another 15 images. I have taken from 4 images from that just to show here. So we'll be training the model about normal and abnormal microscopic cell and the input images. First of all, as you have already seen in the architecture, it takes the input images 224 cross 224 cross 3, and we'll be passing through the convolution layer, which is 224 cross 224 cross 64, and 64 is the number of filters, and it goes by, after that we will be doing max pooling. So I'll be explaining in details what is the max pooling doing here, what is the convolution, how it is reducing the number, the size, image size, the height and width, and what is the work of here. It is just an elaborate explanation of the architecture, which I have already previously explained, that how many convolution layers are there. Here you can see that, here you can, as we have already discussed, that there are 13 convolution layers. So you can count up to that 1, 2, 3, 4, 5, 6, 7, 8, up to 13 convolution layers are there, and 5 mix pooling layers. And as you can see, the image height and width is also reducing one after another. So at last, after max pooling, 2D max pooling here, it gets flattened. That is, there is also a layer called flattening layer. And how this value came, that is multiplying the filters, this value comes, and then we'll be passing it through fully connected layer, where another name of fully connected layer are dense layers. So there are 3 dense layers that will be passing through. So, and what is the work of dropout layer? Dropout layer generally is used to reduce the risk of overfitting. Like we can also skip for normal classification architecture, but VG16 is somehow is a larger model. Like dropout layer is much needed. Otherwise, there is a chance of overfitting. So we have there dropout layer is also included, and after the fully connected layer, here one means that from the two binary classes it classifies a single image. So the target cell here it has classified as abnormal macroscopic cell. How it classified? That is the test image we will be giving in the code. If we take that as abnormal microscopic cell, then it classifies correctly as abnormal macroscopic cell. While in the code, when we will be testing the image, while in the code, will be when we will be testing the image, giving the normal microscopic cell, then it also classifies as normal. So and I'll be showing the code that is I have used. So you can see that I have tested giving both normal and abnormal cancerous cells, and my model, that is VGG16, classified accurately. Proceeding towards my next slide. So this is just a normal data augmentation part which I have explained. Like what is data augmentation? It is the process of increasing the amount and diversity of data. Like when do we do augmentation? When we have limited number of images in our data set, and we need the model to learn more accurately and precisely, we just augment the data. That is, we do not collect new data. Rather, we transform the already present data. Like I have for here in AGMC2 dataset, we have limited number of images. For abnormal microscopic cell images, we have 50 images, and for normal, we have 50 images. So it is not enough for the model to learn what is actually the abnormal and normal cancerous cells. So here we will be performing augmentation. Like what are the operations that are performed in augmentation? There is rotation. This is just an original image of an abnormal microscopic cell, and if we perform rotation, this look like this. There is rotated image, and shearing that looks like this shared image, and zooming, cropping, flipping. These are some of the augmentation operations that we generally perform for our data set to become like more for the model to train. We can increase the amount of images by doing augmentation, which is generally necessary, otherwise our model could not learn. Since the dataset, that is AGNC2 dataset, contained a limited number of images, so different augmentation operations were performed in both the datasets. That is, I will be using high-level dataset afterwards. So augmentation operations were performed in both datasets to increase the dataset size and enhance the model performance. So these are some of the augmentation techniques which I have performed in my code. So now, as I have already said, I have given an explanation of my convolution operation, how convolution operation works. So let us take an abnormal microscopic cell, and when we will be performing convolution over that, it is just a small portion of the image. If we take this patch and it consists of numeric values, as I have already considered an image earlier, where we can see from the CAD there are, when we enlarge this consists of numerous numeric values. So this is just like that. Let us consider a teeny patch from that image. Sorry, image is written over there. So I is a matrix which is 5 by 5 matrix and a 3 by 3 edge detection filter. This is a 3 by 3 edge detection filter to detect the edges, because in this type of cell the edge detection is necessary. So that detects edges. When we apply convolution filter to an image, the output feature maps height and width are determined by this formula. That is the W minus F plus 2P by S plus 1, where W is the input size. That is, what is the input size of this image? 5 by 5 and filter size is 3 by 3. Padding here will be doing no padding, so it will be 0. And stride, since the how many pixels the filter moves each step, it will be 1 since the filter is moving one step at a time. So just putting all these values, the output size is 3 by 3. So the output is 3 by 3 for both width and height. I mean, for next slide, you can see here when I'll be multiplying the filter, that is convolution, doing on convolution operation over the 5 by 5 matrix with the filter, then it comes like multiplying the values 1 into 1 plus 2 into 0, and in this way I'll be getting the value minus 6. For output 1 1 it is minus 6. In similar way for output 1 2, I mean moving each times the filter moves one step ahead, and in this way it moves 9 times to cover the whole 5 by 5 matrix. So because the output will be a 3 by 3 matrix, so it have to cover 9 times. Like if the here the output for 2 1 is minus 6, 2 2 is 10, and 2 3 is 10. Proceeding towards next part, and output 3 1 is also minus 6 and 4 and 14. So output feature map would be like I'm taking all the 9 values here and minus 6, 4, 14. So here we can interpretation or the summary comes like that. The negative values, that is the convolution operation, will be negative values for output and positive values for output.
Joydeep, please see the issue. Joydeep.
Abadi गेलेला. Technical issue again came up. The network is very slow here today. So continuing from the previous part. Am I audible? Okay.
Yes, yes.
Yes, audible.
Okay, sir. So I have performed a convolution operation. As I've said, the output here comes minus 6, and for output that is this place, for this part, the output is 14 and 14, and collecting all the values all together in the next slide, the feature map comes like this. Just I've taken all the values. How this works is just normal multiplication, like 1 into 7 plus 0 into 8 plus minus 1 into 9 plus 1 into 1 plus 0 into 2 plus 3 into minus 1, and for 6 into 1, 7 into 0, and 8 into minus 1. In this way, we have performed convolution operation with the filter. That is the edge detection filter. So the output feature map looks like this, and the interpretation, that is summary, we can say that the negative values means the left edges are from, that is from the image till now after the convolution operation. Our model predicted it like that the left edges are dark to light, and high positive values means there is a strong edge. Since it here we have performed convolution operation with the edge detection filter, so the high positive values, that is the 14, here the highest positive value is 14, whereas the negative value is minus 6. -6 generally represent the edges from dark to light, and high positive values, that is there is a strong edge in the vertical direction, and this 3 by 3 filter is acting like a vertical edge detector, like a Sobel filter. So for next slide, I guess you have understood how convolution operation works. So we'll be proceeding towards ReLU activation function. What is ReLU? After convolution, we apply ReLU, that is rectified linear unit. It is generally used to remove the negative values. Like this is the formula for ReLU, that is rectified linear unit. It just removes the negative values and to zero, that is converts the negative values to zero and keeps the positive value as it is. Here the output feature map that we have got from the previous slide is -6, 14, 14, -6, 10, 10, and -6, 4, 4. So converting after applying ReLU, we get the -6 get converted to 0. Here all the negative values converted to 0 or positive values are greater than 0. Like the negative values after applying ReLU just converted or gets positive, that is converted to 0, and the positive remain the as usual. So coming towards the pooling operation, that is in VGG16 architecture, after convolution operation and ReLU, we get the max pooling, right? As you have already seen. So what is the work of pooling operation? Pooling reduces the image size by taking the maximum value in each small region. Like from each and every region, it takes just the maximum value. Formula of pooling is given by max of a particular part of, and that is, we know the ReLU feature map is like this from previous slide. That is the output. So top left region, that is first part, that is this part. From this part, we can say the maximum value is 14. That is, if we see this first 2 by 2 part, then we get the maximum value here 14, because in pooling operation it just takes the maximum value from this 2 by 2 matrix, and from this part it is maximum 14, and here the maximum 4, because there are the values are 0, 10, 0, 4. So maximum here comes, that's a mistake, it will be here, it will be considered as 10, and from this part its maximum value is 14. So 14, the 2, what is the 2 by 2 pooled map? It's 14, 14, 10, 14. So what is flattening? After several convolution and pooling operations, the pooling operation reduces the image size by taking the maximum value in each small region.
Joydeep, please see the issue.
Yes, a technical issue came up just now. So I'll be continuing from this part. What is flattening? As you have already seen, what is convolution, pooling operation, and then ReLU activation function. Now comes the flattening layer. After several convolution and pooling layers, we get a 3D small 3D feature map. Flattening means converting one long vector to pass it through the dense layers. Hence, flattening is an important step. Considering the 2 by 2 matrix that you have already found in the previous slide, that is 14, 14, 10, 14, while after flattening this, it gives us like a flattened vector. Then it will be passed through the fully connected layers. And what is softmax activation function? The softmax activation function is VGG16 is the specifically in the final layer, that is after the dense layers. It will be used. It is used for multi-class classification task. Um, Here is how it functions. Output layer transformation. After the convolution and fully connected layers, VGG16 model produces a set of raw unnormalized score. That is, the scores are not up to that level. They are also known as logits for each of the possible output classes. The softmax function takes these raw scores as input and transforms them into a probability distribution over these classes. This means that for each input, the softmax provides a probability for each class. There is what is the work of softmax activation function? It just provides a probability. This is the maximum chances of that particular image that what will be passing has the probability that is it can belong to this class, and that is it gives a probability about that, about how much likely is that input belongs to that particular class. So code running platform for AGMC, I have used Google Colab. There are various platforms generally to run deep learning code. For this data set, I have chosen Google Colab over other platforms because it is a cloud-based environment that comes pre-installed with many machine learning libraries. Also, the primary reason provides free access to GPU and TPU resources that significantly accelerates the training process. Since it is a broader classification architecture, so I took the help of GPU using Google Colab. So that's why I preferred Google Colab over other platforms. This is the code that I have used to classify the binary class segmentation to classify the AGMC2 dataset, that is the normal and abnormal cancerous cells. So first of all, it's nothing, just importing the required libraries from scratch, like import what are the libraries that needed to be imported. So models import, importing image data generator. Image data generator is a keras utility function like that helps to load, pre-process, optionally augment images. That I have already explained what is data augmentation. It helps in augmentation. And then import Adam optimizer, import matplotlib.pyplot splt for plotting. And so these are the directories for training and testing images. That is, we have to define these directories in Google Drive. In Colab, we have to upload the datasets in Drive, and then we have to access them. That is, the path is given from that part. And this part is the image or augmentation. As I have already explained, the operations in image augmentation consist of rotation and then zooming and then flipping. So these are the some of the augmentation operations that I have performed just to increase the number of images or increase the number of cells, that is abnormal and normal cancerous cells. So this part is considered as training and training gen flow from directory. That is, what are the directories? These are the images. It just load images from the training directory and apply augmentation. Here the class mode is binary because I am using a binary classification data set. For a higher level data set, that is multi-class, it will be categorical. And I have taken the batch size as 32. You can consider it provides the best output. So I considered batch size as 32. It can be reduced or can be increased. So proceeding towards my next slide, this is the whole VGG16 architecture code. Like first of all, we have defined VGG16 and what is the input shape. As you have already seen in the architecture, the VGG16 takes input as 224 cross 224 cross 3, and the number of classes is 1 because it's binary. You can give 0 or 1. I have considered 1. Input shape, that is block 1, block 2, block 3, block 4, block 5, because there are 5 convolution operations in the architecture. So I just in first there are 2 convolution architectures, and then max pooling is done, and similarly the filters are also increasing. You can see from 64 it became 128, and then 256, 512, and after max pooling, the fully connected layers comes, and after flattening, dense layer dropout activation function, as you have seen in the architecture. I have already used that, and then after dense, after dropout, then this is the whole model summary. This is the whole model summary. The output comes like that of the whole model summary, and you can see what are the layers and output shape. What are the shape and the output layers? Here it is described all. So proceeding towards my next slide, there is we trained the model. How much epochs we gave? 30. I mean, I have also trained the model in 50 epochs, 100 epochs, and 200. But the best results came in epoch 30. So yeah, I have considered that in my code. Final training accuracy and printing the final training accuracy comes like this. That is the training accuracy, and my testing accuracy comes like this. So this is the plotting of my accuracy, that is the graph for, and training loss graph is like that. It's just nothing, the code is just to plot the accuracy graph. Proceeding towards my next slide, how to test the trained model in the single image. Like this is the code where I have taken an image. Here you can see the model path. I have given the path, and that is how much, I mean, I have trained the model in 30 epoch. So it is the weight of the model path. That is, I have saved the model, so it is just the weight. And loading, this is the code, load model is just to load the model. And image path, here you can see that I have given an image from the test dataset, which I have not used for training. So in train, there are 2 folders in AGMC2 dataset in both abnormal and normal images, test and train. I have not used the test images for training. I have just uses the train folder. So here I have given the image from the test folder, and it easily classifies it as normal. Like in this experiment, we selected an image from the test dataset which was not used during training. So it considered of normal and abnormal images. The image chosen for evaluation belongs from normal category, and it correctly classified. So similarly, taking an image from the abnormal category also correctly classifies it as abnormal cancerous cell. So yeah, the AGMC2 dataset part has been correctly classified by the model. So proceeding towards the high-level dataset, I'll be continuing this a bit fast, because okay. So there are 7 classes. I have already mentioned in high-level dataset. There are 7 classes: severe dysplastic cells, normal superficial cells, carcinoma in situ, and moderate dysplastic cells. Normal. These are all the cancerous cells that belongs to different classes. So since it is a multi-class classification data set, so we'll be passing in similar way, and it just our model just captures the particular data set which will be giving in test images. So here in high-level dataset, the total number of images are 917. In training, we are sending 643 images. For testing, 274 images. In training, these are the classes, the same classes are there for testing also, but these images are not, these 274 images are not trained. These are just kept for test data, just to test if the model learned correctly or not. So I have used Kaggle here instead of Google Colab, because Kaggle provides much more GPU access than Colab. So yeah, Kaggle is highly beneficial as it comes with the free availability of GPU, being a major advantage for deep learning task. And this is the code that I have used. There's a similar code as the previous one, just it's multi-class data classification data set. Here I have given the training directory and testing test directories, and the code is same. Just here something will change. Like here the class mode is categorical, and for that dataset the class mode was binary. And the number of classes here will be 7. Here you can see the number of classes is 7, and for that part the number of classes was 1. So these are the small changes that needed to be done while performing this while training this code. And this is the model summary. So yeah, the training accuracy came 89% and the testing accuracy came 79%. This is the plotting graph. So testing the trained model in the single image from 7 classes. That is, I have taken an image. Here the image path you can see from Harleb dataset. I have taken the image from the light dysplastic class, which I didn't use in train. So it predicted correctly the light dysplastic class. In this experiment, we selected an image from the test dataset, which was not used during training. The test dataset includes these categories. The image chosen for evaluation belongs to light dysplastic class from the test folder, and the trained model correctly classified it as light dysplastic, demonstrating its effectiveness in distinguishing the 7 classes. So yeah, I came to my conclusion part. That is, my model accurately classified both of these images from different data sets. In this work, I have focused developing deep learning model for image classification tasks, covering both binary and multi-class problems. Main objective was to accurately classify cervical cell images in well-known data sets, that is AGMC, TU, and Harleb dataset. For AGMC-TU, it differentiated between normal and abnormal cells that helps to identify the signs of abnormality at an early stage, while on the other hand, for Harleb dataset, which consists of 7 distinct cell categories, our approach was to able classify each cell image into correct category with high accuracy, and it correctly classified. Overall, the results demonstrate that our models can effectively assist in automating the diagnostic process and contributes towards faster and more reliable cervical cancer screening. These are the references I have used, I have mentioned, and this is the whole VGG16 paper I have mentioned that was first proposed by Visual Geometry Group, reference number 9. So yeah, thank you.
So, okay, uh, thanks, Jagrita, for this assistantship of the very basic VGG net. Please, any kind of question from the participants? So, due to power cut and the internet availability, today's session a lot of problem all of you faced. So, I think tomorrow or in the next session, this problem will not occur. So if any kind of not any questions, so we'll go into the next session. Thank you to all. Thank you, Jagrita. Okay.
Thank you, sir.
Everyone, go to the next link for the next session. We will start soon after 5 minutes.